today what I'm going to do is focus a little bit more on the downstream, the RNA-Seq portion of this. But we'll certainly go through the Novo Transcriptome kind of workflow and output. And I'm going to show you some specific ways that we can analyze this output that I think most of you are going to be interested. And I can see from the names on the list, I've spoken with many of you already. We've had lots of people trying out the Novo Transcriptome assembly. There's been lots of great questions. And so hopefully I can answer quite a few of those today. And after this webinar, I'm sure there'll be more questions, and you know, I'll continue to work with several of you on your projects and produce some great results. So for those of you that are new, I'd like to give some of the same background here that we did previously. So just to give you a little bit of history about the company. I'll try to make this brief because we've got a lot of stuff we'd like to cover. But so DNASTAR has three main areas: molecular biology, genomics, and structural biology. We have a software package that covers all these different aspects, or we can have different sub-packages that focus in these different areas. And so, of course, today we're going to be focused on genomics, and in particular transcriptomics. And some background, you know, we've been in this business for over 30 years. We are founded by Dr. Fred Blattner and John Schrader, who are still actively working at the company and coming up with great new ideas on different things. And in fact, the algorithm that we're focused on here was really developed by Dr. Blattner. So this is something that he brainstormed and we implemented in a relatively short amount of time in the past few months. So our software is widespread. You can see it worldwide. We have support in the U.S. and Europe and in Asia. We have great tech support. You can call into our office. You can chat. You can email. I do personal webinars. Look at customer data sets, so I'll help work through any issues that you have with your data. So it really separates DNASTAR from our competitors, their ability to support our software. So we've been leading in the industry since 1984. We have all different solutions, ranging from Sanger to NGS data. Software runs on, Windows and Macintosh. Our assembler also runs on Linux, and we also have cloud options. We're frequently cited in peer-reviewed journals, more than twice as many citations and references as our next nearest competitor. So this really is software developed for researchers for research purposes. So kind of our statement here is providing software for molecular biology and genomics and structural biology, and that just sums up what the DNASTAR Lasergene Suite is. And of course, there's different programs within the suite, and we'll focus on just a handful of them today. Molecular biology, just to give you an idea of what some of the capability is, things like multiple sequence alignment. We can also do sequence editing, virtual cloning, primer design, sort of the basic tool sets. Structural biology, we have some really excellent new tools for protein prediction, for folding, and we're going to be developing some new tools here as well in the next year or so that can further enhance our ability to analyze 3D structures. So some really nice new developments there. And in genomics is where I want to focus today. And I've had several webinars, in the last couple of years on all different topics, and today we're going to really focus again on transcriptomics, but there's multiple different programs here that offer different capabilities, and we're going to dig in a little deeper into our ArrayStar program that is used for analyzing gene expression. So the software then is very adaptable in terms of the different workflows that we can do and the different types of sequence data that we support. And what makes DNASTAR unique is that we can handle, things like de novo transcriptomes, which are very challenging in terms of the size of the data sets, and then turn that around and go into RNA-Seq. We're going to focus on non-model organisms, which can be more challenging because you may not have annotated genomes. You may only have partially annotated mRNA reference sets. So there's a lot more challenges working with the non-model organisms, and that's where you need the flexibility of a DNASTAR software to allow you to adapt and work with these non-model organisms and produce results that you can analyze. The software platforms, of course, Illumina is the main source for transcriptomic data. Illumina is the only one that produces enough coverage across the transcriptome to find those low to moderately expressed genes. And so it's typically Illumina, oftentimes high-seq data that's produced in very large quantities. We'll also start to see some long-read technologies in the future, and we are looking towards nanopore and developing algorithms to support nanopore. And of course, there's support for PacBio and Ion Torrent as well. But today it's going to be focused on Illumina data. The hardware that you need, this is somewhat flexible depending on exactly what you're doing. If you're doing big de novo assemblies of transcriptomic data, you want to have plenty of space on your computer. And what I mean by that is the ideal setup is to have 3 different hard drives. And the way that I describe this is that your computer C drive typically will run the algorithms and run the OS. And you want to have about 32 gigs of RAM on that computer, 4 or more cores. So most of our computers have 6 cores, 12 virtual. So it's a pretty standard desktop for the de novo assembly. You can also use laptops for some of the smaller projects. So if you don't have a big, powerful desktop, you can get by with a laptop, provided you have another disk that can process data. So we call this a scratch disk, and this is key for the performance of our software. And the scratch disk can be plugged in externally through USB 3 or eSATA, or it can be another internal drive in a laptop. Sometimes you can swap out a DVD-ROM drive for another hard drive. And it's really this drive, the performance of this drive, and the speed of your processor that impacts performance more than anything else. And so it's just something to keep in mind. What you don't want to do when you're trying the software out, don't have your C drive do everything, scratch disk for temp files, storage. The typical C drive doesn't have the input-output that it needs to handle that much data. And if you have specific hardware questions, certainly contact me and I can help you there. There's also cloud solutions. So if you just don't have the hardware, you can do assemblies up on the cloud. There's different advantages. I can run multiple assemblies simultaneously. I can store my data. I can easily share it with colleagues because it's up in a safe, secure location. So that's a great option if you're collaborating. Accuracy, we've got lots of literature here on how accurate our assembler and our aligners are. This is based off of human exome data sets that are the most curated data set that's available. So across thousands of variant locations, we can find those variants at about 99.7, which is what we believe is the highest in the industry. So we think we're right on the leading edge for accuracy, and we're always striving to improve there. And again, I'm going through this quickly because this isn't quite the group of people interested in exomes, but it is reproducible across 3 different exome data sets where our sensitivity is well over 99%. And that same algorithm, though, is used in the other workflows. So what's the advantage here is that you know that at its core, our aligner is going to produce accurate results even in non-model organisms. So you can refer back to this kind of an accuracy white paper and comparison to have confidence that the software is in fact putting things together accurately. So today we're going to focus on the new workflow, which is the Novo Transcriptome, which then can be turned around and used for RNA-Seq. So I'm going to demonstrate that. So I'll jump out of the PowerPoint at this point. And actually maybe I'll stick with it. I'll show you just a couple. Actually, let's stick with it. We'll rather than come back to these later. So and this is what I covered in the previous webinar. So just as a kind of a recap, de novo challenges are that there's millions of reads. Even the smallest de novo non-model organism data sets have 50 million reads. A lot of them have 200 to even 500 million Illumina reads. And so that can be very difficult to de novo assemble. And so we develop new algorithms that can handle those data sets on a desktop computer, right, and do it relatively quickly. And then on top of that, it can create a reference mRNA set for those novel organisms. And that's the key part. The key point is this third step is actually assigning annotations to those contigs that can then be used for RNA-Seq. And rather than just having contig names 1, 2, 3, 4, we're going to have some idea of what that gene is and get an identity to that gene. And so that's really the key, is crunching through all the numbers, getting an annotation, and then using it in the RNA-Seq. The performance, it's been excellent for us. The assembly time, roughly 12 hours per 100 million input reads, and it's a roughly linear correlation between input data and assembly time. And then the number of transcripts, of course, seems to saturate. Once you get to about 100 million reads, most transcriptomes, you get almost everything you're going to get. You pick up a few more rare transcripts as you increase data. But if you're in that 100 million read range for your input, you're generally getting most of your transcriptome. So today, we're going to look at a data set that I downloaded from the Short Read Archive. It's a Nile tilapia, and it's just one I picked randomly. We've got many different data sets that we use to test, and this particular data set has 3 tissue-specific Illumina HiSeq runs. So it's about 6.5 gigabases of total data. And just to give you what some of the de novo transcriptome results are, in this data set, it's about 60 million reads, and about 80 to 90% of those assemble. The assembly time is about 13 hours on this one. Annotated transcripts, we get 27,000, and we get about 500 novel transcripts that do not hit any annotations in the database. Now I'll jump out of the PowerPoint. So that's the data that's been de novo assembled. And what that looks like then, I'm going to go back here to SeqMan Engine. I won't go through that whole, we set that project up before. So at the end of that workflow, so again, in the previous webinar, we set up the assembly, and we got contigs, and those contigs could be identified or novel. And I'll just open this table up, I think, is similar to where we ended before. And so what we get then is a table that's interactive with the individual assemblies. So this table you can see says identified transcripts 27,408, and novel transcripts is going to be around 500, so 561. So after that de novo assembly is done, we get this package of output, and each one of these rows then, if I double click on one, it will open up a small editable SeqMan project file. Right, so I can go and look at the data. I could edit this if I wanted to. I could export a consensus, save the consensus out. So that's different things I can do with these transcripts. And there's all this information from the de novo assembly. So if I look at this row that's highlighted, there's a gene name, there's an organism name. So this is the Nile tilapia, and I get the accession number. This comes from the database hit. I'll keep scrolling over here. And I get a description of what the gene is. And there's a lot more information. I get, it tells me what database this matched. And I'll go ahead and show you what that means. So this is the vertebrate other database 83. We get the transcript length, the match of the transcript to the database sequence. And so if I sort here, I can see, and you can see this from the screen, that most of the matches are well over 90%. So it's very strong hits to these, the mRNA sequences that are in the database. And if I keep scrolling, I get identities and e-values and bit scores and that sort of thing. So a confidence then in the different transcripts. And we can see also by organism too that it's not all, so one of the common questions that came up is, should I just, rather than align to the whole non-mammalian vertebrate database, should we use just a specific database to my organism? Should I go and pick just that file? And for most organisms, you'll find that you get more comprehensive annotation if you pick the entire database. You'll see there's other species here that are coming up and annotating our de novo assembled contigs. So there's generally an advantage to using more than just a single database to use related organisms to help annotate those de novo assembled contigs. And so what I mean by this is when we've set up this de novo assembly, I'll just reduce this and go to the output. So here's my data file here, Nile tilapia. this vertebrate other file, 7 gigs of data, this was automatically downloaded from SeqMan Engine Wizard when I set up the project, and it contains FASTA files then that each one of these is from a different vertebrate species. In fact, they may be mixed. If I look inside here, I can see here's a zebrafish. So we have so this is a FASTA file that contains the zebrafish mRNA sequences. And this database file is key. Let me go back in there. And here's a xenopus. And this header, so this is the Refseq mRNA databases. And in this header is useful information. And we can see there's a GI number, the Refseq accession number, the species name, and then we get this kind of a description here, and then a gene name in the parentheses, right? And so this is annotation information, and it's useful annotation information. The difficult part is that it's not completely standard. These vertical bars are pretty standard, but once we get to the description, if we go through the whole database, that's just kind of handwritten. This varies quite a bit within the database, so it's something that to keep in mind, that if you are using this database, or if you have another database of reference sequences, the software is designed to parse out this information from the header and pull that through that we can load into our analysis downstream. So it is this part that's key. And again, this is from Refseq, so our software is hardwired to pull out the relevant information. But if you have another database, this header probably will be different, and that's where we can help you extract that information and pull it into your analysis. So what I'm going to show you with the RNA-Seq is how we do this, how we actually use this information then to enrich our RNA-Seq analysis, where it's really useful to know the organism, and it's useful to know the description of the genes from our de novo assembled contigs. So that's kind of where we connect the de novo workflow with the RNA-Seq. Okay, so let's go to the RNA-Seq. So again, if you didn't participate in the webinar on de novo transcriptome, it's recorded, it's on our website. I encourage you to watch that webinar, and then this webinar is kind of part 2 of that same kind of a workflow. So I'm going to go back here to SeqMan Engine now and show you how we set up. So we've got the output file, which is the de novo transcriptome. I guess I didn't show you what that. So if I go back here, so into Nile tilapia, it's this folder is the output. So when I do my de novo assembly, I get this .transcriptome folder, and there's lots of data in here. I get a little report file, and it tells me, just a quick, how many transcripts did I get, how many identified from the mRNA database, how many novel. And then I also have, in this reports folder, this all transcripts.search results table. This is the table that we're actually viewing. I can look at this in Excel, and this is actually what we're loading in SeqMan. This multicolored table is that table, and it interacts with all the assemblies. So those are the 2 key components. And we also have the transcripts file. It's a third piece. These are the identified transcripts. These are the novel. So if I look at identified, these are my de novo assembled contigs, and this header of information was extracted then from the database sequences. So we get the accession number, and you can see in this case there's a gene name. So it is this information in the top that allows us to pull and link up the annotation information from the original annotation file. So these are created automatically from the de novo assembly, and they're all kind of bundled in this package, right? And the package is large and contains a huge amount of information. So now when I pick my RNA Seek workflow, the idea is I'm going to align my data back to that annotated set of reference sequences, that 27,000 reference sequences. And so now instead of de novo, I pick templated assembly. Temporary file location, again, that is my scratch disk. So that disk has to be really dedicated to handling these temp files. Has to be fast, lots of space. You want at least a terabyte of free space on it. This one's got 4.3, can handle really big data sets. And now input template files. So I could go and just pick those FASTA files, but really what I'm going to do here is just add a folder. So this is a key piece of information, is I'm going to add this .transcriptome folder. And so the software then will look inside and see all the information regarding the novel transcripts and identified transcripts. And it will use those now as the reference sequences for the RNA-Seq. So I'm adding folder there, and the software will pull out the information it needs. Now I can load the data in. And so this is again, it was 3 Illumina runs, HiSeq runs, paired data. It's multiple sample. So I click multi-sample. I can auto-name here and get pretty close sometimes. And I might say, well, that's good enough. Or if I have more information, I might say, well, I know that these are tissue-specific. So this particular experiment has diverse tissue types. And so if I rename, I can tell the software how to group these together. Something like that. And so what I've done is I've set up 3 different RNA-Seqs at one time. There's also capability to do replicates. I don't have a data set for that, but if you have replicates, you can set this up as replicates and then use the statistics downstream in really an RNA-STAR for gene expression analysis. These are also going to be run as separate assemblies, but it's linked together, so I'll show you at the end here. So I set that up. You may or may not do SNP calling. I can turn SNP calling on. The normalization is going to be set to RPKM. There are some advanced options. I won't dive into those right now, but it's just, here's the basic parameters. And if you're a current DNASTAR customer, if you've owned our software, you'll notice that we've really moved more of the analysis options into the SeqMan Engine Wizard. The RPKM used to be downstream. So what we're doing is consolidating the workflow, streamlining it. And so you make the choices up front, and we can start doing our analysis more quickly is really the idea, to take out some of the steps in the middle. And we've done a pretty nice job with that so far. I'll just name it tilapia. And so you can see I give a project name, pick an output folder, and the output now is going to be 4 different output files. I'm going to have different dot assemblies for each for each data set, tissue specific. Also an RNA-STAR project. So all of these 3 projects are going to get loaded automatically into RNA-STAR where we do our gene expression analysis. I could also do wiggle files. Those are typically only done when we are doing alignments to genomes that have, browser, that can be loaded into browsers. So I'm going to turn that off. But it's useful if you're doing alignments to genomes that you intend to browse. And when I click to next, we see we have a script that's ready to go. So when I click select start, you can watch as each of these assemblies they'll go in a series. And you can kind of watch as they progress through. When it's done, it'll look like this. So I ran this the other day. And so when the assembly is complete, we get this project report in the wizard window. And I can see this first is the I data set. And I get some information here. I can see how many, how long it took, 12 minutes. So, really fast time. There's 27, almost 28,000 reference sequences, and the sequence count is about 20 million in this I data set. And then I can see how many assembled, about half assembled. And as I go to, and we can analyze, so a question will come, well, why did only half assemble, or, whatever the number is. And RNA-Seq data sets, especially from non-model organisms, vary a lot. Sometimes there's a lot of ribosomal. Even though it's been ribo-depleted, we'll see a lot of ribosomal RNA present, sometimes genomic, and they vary a lot depending on, how the samples were prepped. There I can see the liver and the muscle. So I get a report then of the 3 different projects. Now, if my goal is to compare them, by gene expression, there's a button here that allows me just to launch this directly in RNA-STAR. And so I can start my gene expression analysis quite quickly. There's a little bit of new interface on RNA-STAR as well, and you can see these trying to get our attention with a gold star and large font. Would I like to begin to find gene sets of interest? And for those of you that haven't used RNA-STAR, really the key to using it successfully is creating sets and comparing those sets. RNA-STAR is going to pull in all the information from the assemblies, so there's lots and lots of information, including stuff that you want to filter out that's just either low-level signal or noise, and I want to start comparing quickly. So if I click this link, we get a little mini wizard that comes up that asks me how I want to compare my experiments. And so some experiments are just individual, and I want to compare them, or sometimes we have a baseline, like a time course, and I want to compare against the baseline. Sometimes we want to do pairwise with some stats. For this particular setup, it's tissue specific, so I'm going to compare them individually. And so we just kind of step through, and what this allows me to do is give me at least one threshold. And what I'm doing is I'm going to create a gene set for each one of my experiments. And I'm going to say only show me the genes, create the sets with genes that have enough sequence aligned to it that they're worth comparing. And that's subjective. It's typically in a positive number on a log2 scale. So for RNA-Seq, most publications it's log2, log4, once in a while a little bit less than log2. And so this is just kind of an auto set creation. I can do this manually as well. I can go through and apply filters all different ways. We've got lots of different webinars where we go through that in detail. But this is a quick way to say, just make some sets for me. And let's look at this in a traditional pattern. So this is, again, it's very different tissues. So in this case, the transcriptome, they don't overlap that well. It's not, if we did whole organism here, you get a much more similarity between the 3 different sets. But because these are specific tissues, we're going to have many different genes that are expressed in one tissue, not the other, and certainly different isoforms expressed in one tissue versus the other. So we get this kind of, you'll see that the Venn then shows, while there are a lot that have in common, each tissue type, so here's liver, has a subset of mRNAs that are expressed just in that tissue type, at least above the threshold that we picked. So the way that we use RNA-STAR then is we can make comparisons like this if we're interested in the genes that are in common between all, or the genes that are unique, and we can look at the data in different ways. So I can select a region and go to a scatter plot, and you can see just in the scatter plot, what's highlighted is just, those are just the genes that were expressed only in liver. So I can go, again, back and forth, try different quadrants. So the selections then are, and I can change the coordinates here if I don't have. So now we have muscle. Those are just the subset only in muscle. So when I select these regions, it updates. And so the views then, the different ways to view the data are linked together in RNA-STAR, which makes it a really nice analysis tool. Go back to my Venn. So if I picked a certain tissue type, I can also go and say, let's look at a table of those genes. And so this is the gene table in RNA-STAR. You can see we have the gene name here. I'm just going to sort this. And show only the selected genes. So this is a multi-state table. So RNA-STAR, we can control the states here by this pull down. Show only a set, selected, show all the genes. So this is just that subset from liver. And let's see, I'm going to go to the manage columns. So there's a column manager that allows me to also to pick in different values to populate this table. And so you can see there's not a lot right now. I have the name, This comes from the annotation, and I'm just going to bring in some signal. I like to look at just raw counts if they're in a reasonable scale. And you can see that we have this gene name, ACER1, ASCI5, and when we get down to CL, we get these that all look kind of similar. CL are the novel transcripts. So in this data set, there was 561 transcripts that didn't get an annotation assigned. This name comes from the hit to the database, right? So that's how we're able to apply a name to these novel transcripts, or to these transcripts from this novel organism. And when I scroll through, you can see that there's a pretty good subset here that are novel, but not, 10%. And then we get all the genes that are less informative, LOC, a locus name, which aren't as informative. Right, and we get a few more that have the typical kind of gene names. And if I go back to the Venn diagram and look at the intersection, I'll go back to this will just bring more in. So now we have the larger set of mRNAs that are expressed across the different tissue types. And now we can see we got quite a few gene names. So what's nice is we got a lot of different gene names here. A lot of annotations came through. And I'm just going to scroll down so you just get an idea. Out of the 11,549, we've got all these different gene names. Now, I can bring in more information as well. And this is, again, part that for a lot of you that have been working in this workflow already, it's a little piece of information that I can show you here. So if I want to bring in some of the other headers from the FASTA file, so when we look back at this, this is the original information from the GenBank file. So we have these species names and descriptions that are hard to drag through just because they aren't quite standard enough. But we have a way to import them into RNA-Seq, and it uses a kind of a custom import wizard that allows it to parse information. And so this information was put into our all transcripts table. And so I can do a file, and you can do this for any kind of data set. That's in, a tab separated, a comma delimited format. And let's just go to, so I'm going to go into my .transcriptome and grab that report. And I'm just going to, so I show all files. There's my .search results. And this will get smoothed out a little bit in some of the future builds where these steps can be a little bit more automated. But I can bring this in, or I could bring in other kind of annotation files. So again, if you've got annotations in some other format that's reasonable, we have a reasonable chance of parsing, you can try this import wizard to try to pull those in. And so this one is relatively easy. There are easier ones, but it's relatively easy. I'm going to open that. What happens is we get a custom import wizard. And so it recognizes that this is a tab delimited file. And now I can see what the columns are, and I can look at it and say, well, is it tab or space? What works the best? So you can or you can do an other, and I'll show you an other in a bit. But this is tab delimited. We have the assembly ID, which is a conglomeration as the data flows through our software. But then I can see, gene name is parsed out, and I get the organism name and a session number. So that looks pretty good. And here's the description, which is what I really want over here. And it looks like that's parsed out pretty well. So I'm happy with that. So I click next. And now, depending on the format, if it's a tab, table format, there may be headers. And what we see in this line here, assembly ID, config type, that is coming from the table. Those are the table headers. And that header will be brought into RNA-Seq as well. So for example, I can say, here's my gene names. So I'm going to say that's my ID. And I like the organism name. I'm going to call that a description. And I like the accession number. I'm going to call that a description. And I like the description, which is a description as well. And there's more, all this information. I may decide I want, additional information related to how it was annotated, right, from the de novo assembly. So I can call those descriptions, and they'll come into the project. So when I click Finish, the import wizard will pull that data into this project. Now when I go to Add Manage Columns, I can see description, organism name, accession number. And now I have a richer analysis. So now when I'm making comparisons, I get a description of the gene, which organism the annotation came from. And you can see it's not all, again, I just want to reiterate, it's not all from the tilapia. Some of the best annotations are coming from some of these other organisms. And I get a session number, which you can see is not quite parsed quite as cleanly. We get some of these double isoform session numbers here. So for a non-model organism, though, this is extremely useful. Now I can, when I do other types of analyses, having additional annotation information is really helpful. So, for example, I might go and select one of the tissue types and do a clustering. So then I can get look at clustering patterns and zoom in on. I might look at where it looks like there's a lot of differential kind of expression. And now notice as I zoom in, I'll go to the selector here. As I select a region in the heat map, if you look way over to the right, there's a details panel, and the details panel will give me information about that particular gene. What I can't quite show you though is that, see if I can change my scale on my screen here isn't quite letting me see the description. The description is visible down at the bottom, but I can't, my resolution on my screen isn't allowing me to do that without, there we go. So I can see that description that we just imported into the RNA-Seq project. I'm getting the organism name, and I'm getting the description of that gene. So that, again, is new annotation information, and it's pulled into the project. And when I look here if I see a clustering pattern, I may have some novel. These CL genes, again, are novel transcripts. They didn't hit anything in the database very strongly, so they didn't get annotated. But sometimes by clustering patterns, you can have an idea of what that gene might be doing. If it's clustered together with some tissue-specific genes that are doing partaking in a particular function, I might be able to interpret what some of these more novel genes are. And so, again, the clustering and heat maps can be very useful for things like this, tissue-specific, where I'm looking for subsets of genes that are in one, especially in one tissue, but not the other. And again, as I select a region, I can get that detail here on that particular gene. So again, that's some of the advantages to being able to pull those annotations, and I will show you how one that's a little more difficult to pull in. So if I go back to my gene table, and I'm going to show all, and so again I have these novels. I guess we can, these unannotated genes. I'll sort again so we can. Let's see, just show that subset. We'll go to these CLs. So, and this is a question that came up with several people is, well, how do I get, some of these organisms are so novel that you really don't get very many database hits. I mean, that's a possibility. If you've got a very, very novel organism, and even the mRNA searches, if you relax the stringencies even, in the de novo assembly, you still have mostly novel, which is, again, possible. Then you have to have other means to annotate. And, a common way is to do like a blast X. And we don't have that built right now, within our workflow right now. So the way that we do that is, let's see if I have, we go back to this folder. I'll just kind of show you how I had done this. And there are better ways to do this, but so I went back to my de novo transcriptome and went into the transcripts. And it's this novel transcripts file, these 561, right? So I can sometimes just copy that or open it and copy it and do a blast X. And see, I thought I had, yeah, okay. So here's the 561. So I went to NCBI and loaded the sequences in and did a blast X. And in this case, I picked an organism I just picked zebrafish as the organism. If you don't pick an organism and you do BLASTX for the entire database, I don't know how long it would take with 561, but certainly it's going to take a long time. And there are other ways to do this local NCBI database. There's other options that give you more flexibility. And I know there's some more options here that I haven't tapped into fully. But so what I did is I did the BLAST, and I downloaded the table, the CSV comma separated table. And that table then I'm going to import into RNA-Star. And so you can do a BLASTX. Import annotations. And I think this is just sitting. So this is the Danio BLASTX. And now this, the output that I get by default isn't great. You know, it's more difficult to try to get, and you can see the preview is like it's not tab separated, it's not space separated, you know, and of course it is vertical bar separated, which is shift, and I have to put a vertical bar, and that's essentially how these are delineated here. And so I can see that the preview is showing, okay, I've got these CL, my cluster IDs essentially, and in one of the columns, maybe 3 here, it looks like that's as close as I can get to a gene name. So when I click next, so this is kind of how it works, and sometimes you have to go back and make an edit in the file. So if the format isn't quite right, I have to go back in Excel and add a vertical bar somewhere, which is what I did for this to parse out this first column cleanly. I added in Excel and added the vertical bar. But here I can say, well, that looks like my the key here is I need to match one of these columns with what's in the RNA-Star project to get. However you do this, that's the unique identifiers. This name column, that has to match something in one of the rows in that annotation file. If that can happen, then you can assign the descriptions and bring in the descriptive information. Now this file doesn't have headers, so it's not going to be as nice as what we had before because there are no headers here that can be parsed out, but it does typically allow me to parse this in. Sometimes you'll get an error if you've already done it once. And now I've brought that information in, and you can see the header. It doesn't give me that nice description, but it does say, and now I have some information. It's not great, and there's better ways to do it, or I might be able to get some descriptive information if I knew how to use the BLASTX a little bit better. I'd be able to pull in some more descriptions, but that's essentially how we pull in those kind of annotations, and I can manage the columns. So I could say, well, I want that right where I can see it clearly. So there you can see for some of these novel transcripts that didn't hit anything at the DNA level, I can do a BLASTX and at least get accession numbers for some of these. So when I'm doing my analysis, whether it be from a heat map or a scatter plot, I may be able to look up some of these and figure out, what their function is, or tie it in. Have another way to tie this. Bring in some additional descriptors using that import wizard. Okay. Well, I think we covered quite a bit here. I'm sure there'll be plenty of questions. There's all different variations on this workflow. I'll be happy to stick around here. Let's just look at the last couple slides here. I'll just bounce back here. So again, in terms of our software, if you don't own it already, there's all different options in terms of the kind of licenses that you can have. There's desktop network software, institute-wide, cloud software. So feel free to contact me. I can put you in touch with a sales rep, for those sorts of questions. And again, the tech support that we have, it's 7 a.m. to 6 p.m. Central Time. And we have all different training resources, webinars like this that are recorded. We have tutorials, many different videos on specific topics. And again, thanks for your time, and you're welcome to contact me at any point if you do have questions in the future. So again, thank you for your time.
Thank you, Matt. That was an excellent presentation. We do have several questions. If our audience wants to stick around, and if you have more questions, please continue to chat those in. But we'll start with, we have a few questions about the de novo transcriptome assembly step. And so one question was, how were the 561 novel transcripts discovered during the assembly?
So the de novo transcriptome workflow is really a series of algorithms that go through and align the data. And so there's an initial step that takes all the raw input data and creates clusters or contigs out of it. And so a typical transcriptome, you'll have, say, 30,000 discrete clusters. Then there's another round, a different version of the algorithm that compares those clusters to the entire mRNA database. And that's again, automatically imported into the project from SeqMan. And so most of the sequence, in this case with the Nile tilapia, most of them match something in the database well enough, that we could assign an annotation due to that match. However, there are some, and in this case 561, that didn't match anything in the database, even though it was aligned to all the non-mammalian vertebrate sequences. And in that case, if it doesn't match anything, then it gets those sequences, those consensus from those clusters get put into a novel transcripts file. Now, depending on the organism, if you have a very novel organism that just doesn't match anything in the database, you may have a majority of sequences that fall into that category. So that's possible. And then at that point, the next best option is to do a BLAST X and see, is there conservation at the amino acid level? And that is a separate step outside of DNASTAR where we go to NCBI or download a local version and run a BLAST X. So those are kind of the two options for bringing in some annotation information.
Okay, thanks. And then kind of a related question, which you covered a little bit in your answer to the last question, but when you were talking about the known transcripts, you were referring to the gene names. Can you just explain a little bit about where those gene names are coming from?
Yeah, so I can show you specifically here. So the gene names again are coming from the. We'll go and take a look right in the file. So this database then. So this FASTA file. This is downloaded directly from Refseq. So it is the mRNA database for vertebrate other 7. So there's, say, 100 of these databases that contain mRNA sequences for all the different non-mammalian vertebrates. And in these FASTA files, in the header contains the gene names. And it's hard to find it. It's way at the end. It's in parentheses. This FOXC1A is the gene name here. We also have the accession numbers for the mRNA sequences as well. So we have a little bit of information on the gene name and then some isoform information on those specific mRNA isoforms. So when we do the annotation, when we get strong matches to one of these sequences, this is the information that's parsed out, and that includes gene name.
Great, thank you. And then another related question to the database is, can you use multiple databases for this annotation step?
So you can use a different database. So in this case, it is the Refseq database. It has this format. If you have another database from some other source, that can be used as well. However, there is in order to extract out the human readable text in the appropriate fields, there's a computer language that we use to extract out the pieces. It's called a grep language. So if you're using a FASTA file from another source, we can help you change the grep so that it extracts out the right bits of information. What we can't do at this time, though, is we can't combine different databases in one de novo assembly. Because that would require multiple different grep languages at one time, which the software currently doesn't support.
Okay, great. And so now the next couple of questions relate to the RNA-Seq side of the workflow. And one question was, you showed setting up a reference sequence when you were setting up the RNA-Seq project. Is that something do users have to download anything there, or where do those reference sequences come from?
So the reference sequences can be from different sources. So for a model organism, the reference sequence is typically an annotated genome. And then the RNA-Seq data is aligned and quantitated against the annotations, essentially the coding regions in the genome. And there's a fairly sophisticated model for piecing, splicing exons together and finding isoforms and so forth. The other option is a set of annotated or mRNA sequences, so you can use those as a reference. And then the third option is what I showed today, and that's I'm going to use the newly created annotated contigs that I got from my de novo assembly. And so what I showed earlier was loading in a .transcriptome output package from a de novo assembly. And our software looks inside that package and uses both the identified transcripts and the novel as reference sequences. So it's using those consensus sequences for each one of those assembled transcripts.
Great. And then again on the RNA-Seq, is it possible to create replicate sets? And if so, what statistical tools are available for the RNA-Seq analysis?
Yes. So there is in the Wizard interface, there is capability to do replicate sets, and you have to have a minimum of 3. And if you have replicates, then you can do things like a common thing is a t-test. And the t-test will give you look across the samples at each gene and essentially normalize it across the multiple samples. So in some cases you may see a high degree of variability between samples. Sometimes you don't. So I have about half of my customers say, well, they really don't see that much variability, so they don't invest additional in more replicates. They might invest more in more coverage on a transcriptome. But then I have others that especially if they have thinner coverage on the transcriptome. So the thinner the coverage, the more variability you get in the signal. And so if you have thinner coverage, those people tend to use replicates there. And then a t-test is usually the way to go.
Okay, thanks, Matt. I think that's all the questions for right now. At this time, we just have a few minutes left. So I just want to show a couple of resources that Matt mentioned. So in a moment here, you should be seeing my screen. I just have the DNASTAR website pulled up. And from our home page, you can see there's a webinars link. If you click on that, you can get to our webinars page, which in the next few days, we'll have a recording of this webinar down here in the archived webinars section. And just a couple places below that, you'll see this de novo transcriptome assembly and RNA-Seq analysis webinar. That's the webinar Matt referred to that we did last month, and it really covers the de novo transcriptome assembly and annotation steps in a lot of detail. So if you're interested in learning more about that part of the workflow, I definitely recommend you check out this webinar as well. Of course, we'll be having more webinars in the future, so you can always check back and see what other training sessions are available. I'll send everyone who's registered for today's webinar a link to this page, but I just wanted to point it out in case you're interested in looking for yourself. And then in addition, from our support menu, you'll see we have all kinds of resources from online help, written tutorials, FAQs, as well as shorter 2 to 3 minute videos. So if you want to learn about any other features of our software or get a little bit more in-depth view of some of the things Matt showed today, those are great resources for you. If you didn't get your question answered today, we'll follow up with you after the webinar. I think I saw a couple more chats coming in just now in the last few seconds. And of course, if you think of other questions later, feel free to email webinars@dnastar.com, or you can email Matt directly. Thanks everyone for joining us today. We hope to see you again at